Hello, I'm Glenn Murphy with Burroughs Wellcome Fund Science Explorers, and today we're at Durham, North Carolina's Duke University with Dr. Nick Buchler. Dr. Buchler is a biophysicist who specializes in systems biology. That is, how the bits and pieces of living systems work together to perform the essential functions of life. Incredibly, he actually builds new living circuits inside cells to see what will happen as they grow. Dr. Buchler, thanks for joining us.
Glad to be here.
So let me get this straight. You build new living things inside cells to see what will happen when they grow. I mean, how do you go about doing that, and where do you get your parts from?
Right. Well, that's a great question. So, sort of our approach is, my background is physics, and so I sort of take a bottom-up approach to biology. And so what we try to do is, to take a quote from another physicist, is what I can build, I understand. And so we try to actually understand how gene circuits work in living systems, and we do this by building very simple circuits in yeast. And so we take our parts from anywhere from jellyfish, we've got them from fireflies, humans, plants, you name it, bacteria.
And as I understand it, you look at clocks and switches inside cells to sort of see what makes them tick. How do you go about that?
Right. Well, so we take our inspiration from nature, and we actually look at some common threads and some oscillators. So for instance, we humans actually have a gene circuit that oscillates in our head, 24-hour clock. So if I were to put you in the dark, actually you continue your rhythms on 24 hours. And so we looked in nature for inspiration on how those might have been built, and then we take those parts and we put them together and try to build artificial circuits in yeast that oscillate.
Cool. So how did you end up doing this for a living?
Well, I think it comes back that my father actually is a tinkerer and a physicist, and so I started off in physics, but really I think my favorite class that I took in college was biology and learning how genes and proteins work. And that really got me hooked. And so as a physicist, I eventually wanted to understand, from a physics viewpoint, how living systems sort of regulate and work.
Awesome, cool. So would you mind introducing us to your team, showing us around your lab?
Absolutely, let's go.
Great, let's go. So the first thing I notice is you're not alone in here. Are all these fine-looking people here part of your team?
Yeah, absolutely. We've got people from all over the place.
Hi, I'm Tony. I'm a cell biologist from Maryland.
Hi, I'm Angie, a biologist from Colombia.
Hi, I'm Mariana. I am a computational biologist and I am from Mexico.
I'm Sargis, physicist from Armenia.
I'm Hungwon, a physicist also, and from Korea.
Hi, I'm David, aspiring computational biologist from Chicago.
So what's the coolest piece of equipment you guys get to work with?
We have a time-lapse microscope here. We call it Lord Vader, for obvious reasons. Let's take a look at it. We've got an incubation chamber in here. We can keep the yeast nice and warm. We can feed them in real time while they're growing under a microscope. What we do is we take a picture every 3 minutes. We look at sort of the GFP, yellow fluorescent protein, or even the firefly luciferases, and then we take all these images and put them together to make a nice movie and look at how genes turn on and off over time.
So what's the most exciting discovery you've made so far?
Oh, well, so this one was a surprise, and we use green fluorescent proteins from jellyfish, one of the parts, to look at how gene dynamics change in time. And in this case, we used something different. We used a gene from a firefly which produces light on its own, which is very different. And so we've actually got yeast producing light under a microscope.
So you've got one bug lighting up another.
One bug lighting up another.
Awesome, brilliant. Well, thank you very much once again, Dr. Buchler, for having us.
Thanks for coming.